Okay, I'll start today's session now. If anybody wants to join after this, they can join on their own. So we'll start our session. I'll just share my screen and one of you can unmute and let me know if my screen is visible and if I'm audible. Give me a second. Okay. Can one of you please unmute and let me know if my screen is visible and if I'm audible? Anyone? Is my screen visible? Hi, is my screen visible? I can't hear you guys. It seems like there's no issue. Is my screen visible? Okay, you can just write it down in the chat box or maybe you unmute and let me know if my screen is visible and if I'm audible. I'll just move ahead with the class because I think it's fine. Okay, so good evening everyone. My name is Aishi. I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay. And basically, this is the third live session for this course. So what we do is basically we discuss some old assignment questions from this course. Taken by course instructor Professor Aishi Dasgupta, so that we don't not only discuss the questions, we also discuss the concepts behind those questions, so that it's easier for us to solve similar type of other question, right? That is what we do basically in this course, in these live sessions. So I'll start today's session. If you have any questions, you can just write it in the chat box or maybe unmute and let me know. If I know the answer, then I'm there. I answer you. If I don't know, then I'll take time. I'll give you the answer by next class for sure. Okay. So moving on to the first question for today. Oh, sorry. 1 second. Forgot to put it on animation, but ideally it should not show up immediately. Anyway, how many oxygen molecules can bind with hemoglobin? So I know you saw the answer already, but anybody would like to answer? I need your explanation along with it. Anybody? The question is, how many oxygen molecules can bind with hemoglobin? So I need the detailed answer. Anybody? I'll wait for a few minutes and I'll wait for you to come up with the answer. Anybody? I'll wait for a few minutes. If you want to say the answer along with the detailed answer, if you want to explain along with the detailed answer, please go ahead. Anybody? Okay, so the answer to this will be 4. Now we'll discuss why it is 4. So basically, a single adult hemoglobin molecule can bind up to 4 oxygen molecules. 1 O2 per heme iron center. Now, why and how? Adult hemoglobin is a tetramer made up of 4 polypeptide chains, 2 alpha and 2 beta. Each chain contains a heme prosthetic group. At the center of each heme is an iron ion in the ferrous state. Now, each ferrous Fe2 plus can reversibly bind 1 O2 molecule, so 4 hemes. 4 O2 per hemoglobin tetramer when fully saturated. Okay. Now, if you talk about the single chain proteins, by contrast, myoglobin is a single chain protein with 1 heme and binds at most 1 O2. That's why hemoglobin multi-subunit is suited for transport. Myoglobin single subunit for storage. Now, if you talk about the cooperativity and the oxygen dissociation curve, hemoglobin exhibits positive cooperativity. Binding of O2 to one heme increases the affinity of remaining hemes for O2. This produces the characteristic sigmoidal S-shaped O2 hemoglobin dissociation curve. Now, this cooperativity allows efficient O2 loading in the high PO2 environment of the lungs and efficient unloading in the low PO2 environment of tissues. Right? So a protein, hemoglobin is nothing but a protein in RBCs that carries oxygen to the peripheral tissues to maintain cells. It's made from 2 similar proteins, alpha and beta, and both proteins must be present for hemoglobin to pick up and release oxygen, right? So before birth, beta protein is not really expressed. Gamma, a hemoglobin protein found only during the fetal development. Now, how is hemoglobin made? So the blueprint for hemoglobin already exists in DNA. Obviously, that is the genome. An individual has 4 genes that code for the alpha protein or alpha chain. 2 other genes code for the beta chains. The alpha and the beta chains are made of equal amounts, and the protein chains join in developing RBCs and remain together for the life of the red blood cell. This is the structure of hemoglobin. If you see, hemoglobin is consists of 4 globin molecules, 2 alpha globins, 2 beta globins. Alpha and beta. Now, the alpha globin chain is composed of 141 amino acids and the beta globin chain is composed of 146 amino acids. So, both alpha and beta proteins share similar secondary and tertiary structures, each with 8 helical segments. Each globin chain also contains 1 heme molecule. So, heme molecule is composed of a porphyrin ring which contains 4 bilateral molecules cyclically linked together and an iron ion ligand bound in the center. Did anyone ask anything? Anyway, I could see some. Okay, no voice. Okay. Can you hear my voice now? Anybody? Can you just write if you can hear my voice now? Hello, hello. Can you guys hear my voice? Anybody? Hello. I don't know what the issue is. Hello. Can you guys hear my voice now? Can you hear now? Anybody? Guys, can you hear my voice now? So you can hear, right? And my slide was also visible. Okay, okay. Okay, okay. Sorry, sorry. I'm really sorry. Thank you for right. Actually, I have been asking and there was no response. I thought there's not a problem, but thank you for clarifying this, and I'll start the class again. Don't worry. Uh, Somebody wrote that NPTEL YouTube links are yes, yes, yes. I'll upload the. I have uploaded, but there's some issues with because of it. It has not been made public yet. My YouTube links will be uploaded by today or tomorrow for sure. Thank you. So I'll just restart what my session. Sorry, sorry, sorry. Really sorry for the network issue. I don't know what keeps happening exactly. So, as I was saying, How many oxygen molecules can bind with hemoglobin? Anybody wants to answer, or should I go ahead with the explanation that I already started with? Anybody wants to answer? I need an explanation as well if you are answering. Okay, anyway, so I'll just talk about it. So the answer is actually 4. Okay, so a hemoglobin is nothing but a protein in RBCs that carries oxygen to peripheral tissues to maintain cells. It's made from two similar proteins, alpha and beta, and both proteins must be present for hemoglobin to pick up and release oxygen, right? Now, before birth, beta protein is not really expressed. Gamma, a hemoglobin protein found only during the fetal development. So the blueprint for hemoglobin exists in DNA. That's obvious because that's the genome that is coding for everything. And an individual has 4 genes that code for the alpha protein or alpha chain. 2 other genes code for the beta chain. So the alpha and the beta chains are made of equal amounts. Now the protein chains join in developing RBCs and remain together for the life of the red cell. The structure of hemoglobin, if you can see, it comprises of 4 globin molecules, 2 alpha globins and 2 beta globins. Now, if you talk about the alpha and the beta globins, this is the structure. Now, the alpha globin is composed of 141 amino acids and the beta globin chain is composed of 146 amino acids. Now, both alpha and beta proteins share similar secondary and tertiary structures, each with 8 helical segments. And each globin chain also contains one heme molecule. So, heme molecule is composed of a porphyrin ring which contains 4 pyrrole molecules slightly linked together and an iron-iron ligand bound in the center. So the heme molecule is located between the helix E and the helix F of the globin protein. And the alpha and the beta subunits of the globin chains exist in 2 dimers which are bound together strongly. Now oxygen binds to the heme iron ion tightly, and if 2 heme molecules come together in the presence of oxygen, the iron atoms will oxidize and irreversibly bind to the oxygen. And this irreversible binding would not be of use in the hemoglobin molecule because oxygen needs to be released in the tissues. The globin chain prevents this irreversible binding by folding the protein around the heme molecule, creating a pocket to isolate the heme molecule from other heme molecules. The globin molecules allow the iron atom to form loose bonds with the oxygen, and therefore the ability to bind to oxygen and then release it into the tissues without becoming permanently oxidized in the process. Now, carbon dioxide CO2 transport in blood. Mechanism one of what CO2 transport in the blood, like you can see, oxygen binding to hemoglobin CO2, and if it's carbon dioxide binding to the hemoglobin, it's called a carbaminohemoglobin. If it's oxygen, then it's oxyhemoglobin. Okay. So, how does the oxygen transport happen? So, oxygen diffuses from alveolar air into the blood because the venous blood has a lower partial pressure. So, the oxygen dissolves in the blood. And a small amount is carried as solution. The remainder of the oxygen is carried in chemical composition with the hemoglobin in red blood cells. Hemoglobin behaves as a weak acid. Oxyhemoglobin also behaves as a weak acid. Because both forms of hemoglobin are weak acids, the net reaction for the interaction for oxygen with hemoglobin results in the following equilibrium. If you can see over here, I'll just put in my pointer. So you can see some videos and references. So oxygen is carried around the body in RBCs, we know. RBCs contain the molecule hemoglobin which joins with oxygen to give oxyhemoglobin. Now, hemoglobin is globular protein which is made up of 4 peptide chains. Each chain contains one heme iron group. Each heme group can bind with one oxygen molecule O2. This can be summarized as like this: Hb plus 4O2, HbO2, 4. This is a reversible reaction. Now, 100% saturation means the hemoglobin is carrying its maximum amount of oxygen. 50% is only adding half of what it could carry. That's self-explanatory. Now, each red blood cell carries millions of hemoglobin molecules. That's a lot of oxygen. Now, oxygen saturation is the amount of oxygen bound to the hemoglobin in our blood, expressed as a percentage of the maximum. The units are this: so blood leaving the lungs normally at a saturation of 95-99%. Now, an experiment was carried out to measure the saturation of blood with oxygen at different concentrations of oxygen. So, this is called the oxygen dissociation curve, right? Here, the percent saturation on the y-axis and the oxygen partial pressure on the x-axis. Now, the differences in blood color according to saturation with oxygen can be measured with a colorimeter, and this device measures the amount of light transmitted through a patient's finger. This varies according to the oxygen saturation. So, oxygen saturation of the blood is a sign of health. So, normal values for blood leaving the lungs or in artery are 95-99% O2, and lung diseases, smoking, or heart problems may cause oxygen saturation to fall to below 90%. Now, the oxygen dissociation curve can also be affected by levels of carbon dioxide in the blood. Remember, levels of carbon dioxide go up when rates of respiration increase. Now, carbon dioxide dissolves in the blood to form carbonic acid. Now, this change in blood pH affects hemoglobin in the red blood cells, causing them to lose their oxygen more readily. This is known as the Bohr effect. Okay, if you can see. Now, what do you know about the cardiovascular system? So, it's a circulating transport system. There's a pump, which is the heart, a conductive system, that is a blood vessel, and a fluid medium, the blood. Now, the function is basically to transport materials to and from cells: oxygen, carbon dioxide, nutrients, hormones, immune system components, waste products, etc. Now, blood is specialized fluid of connective tissue. It contains cells suspended in a fluid matrix. So, what are the functions of blood? Transport of dissolved substances, regulation of pH and ions, restriction of fluid losses at injury sites, defense against toxins and pathogens, stabilization of body temperature, etc. So, whole blood, we have the plasma fluid, formed elements, all cells and solids. Now, plasma, where there is water, dissolved plasma proteins, other solutes. It's similar to an exchanges fluids with interstitial fluid is matrix of formed elements. So, what are the formed elements? Basically, RBCs or erythrocytes, which transport oxygen. WBCs or leukocytes, which are part of the immune system, and platelets, which are cell fragments involved in clotting. Now, what is the general characteristics? This is the normal temperature or the physiological temperature of blood. It has high viscosity and slightly alkaline pH. Now, we will not go into too much detail. We know about the plasma, which makes up 50 to 60% of blood volume. More than 90% of the plasma is actually water, it's aqueous base. Water, extracellular fluid, interstitial fluid, and plasma. Materials, plasma, and IF exchange across capillary walls like water, ions, small solutes, etc. Now, what are the classes of plasma proteins? Albumins, which make up 60%, globulins, which make up 35%, and fibrinogen, which make up 4%. So, albumins transport substances like fatty acids, thyroid hormones, steroid hormones. Serum is liquid part of blood sample in which dissolved fibrinogen has converted to solid fibrin. What are the other plasma proteins? 1% of plasma changing quantities of specialized plasma proteins, enzymes, hormones, and prohormones. Now, 90% of our plasma proteins are made in liver, antibodies made by plasma cells, peptide hormones made by endocrine organs. Now, total blood volume in liters will be 7% of the body weight in kilogram. About half the volume of whole blood is cells and cell products. Now, how do we measure RBCs? RBC count reports the number of RBCs in 1 microliter whole blood. Hematocrit percentage of RBCs in centrifuged whole blood. Now, RBC in male it is 4.5 to 6.3 million. In female it's 4 to 5.5, a little less. Whereas hematocrit you can see the values. Now, what is the structure of RBC? Small and light, highly specialized disc-like structure, thin in the middle and thicker at edge. They lack nuclei, mitochondria, and ribosomes, and they live about 120 days. So, hemoglobin is a protein molecule that transports respiratory gases. Normal hemoglobin in an adult male will be 14 to 18 gram per dl deciliter whole blood. This is the hemoglobin structure. It's a complex quaternary structure. If you can see from the figure, alpha chain 1, alpha chain 2, beta chain 2, beta chain 1. Four globular protein subunits, each with one molecule of heme. Each heme contains one iron ion. Iron ions easily acid with oxygen, oxyhemoglobin, or dissociate from oxygen, deoxyhemoglobin. Now, fetal hemoglobin is a strong form of hemoglobin found in embryos. Takes oxygen from mother's hemoglobin. So, how do you recycle RBC exactly? It's the process of recycling RBC. If you can see over here, new RBC formation happens in bone marrow. New RBC is released in circulation. Lifespan is 120 days. They get broken down amino acids, heme, and old and damaged RBCs, 10% hemolysis. They go to the hemoglobin go to the kidney and is eliminated in urine. Now, what are the components? Building red blood cells require amino acids, iron, vitamins B12, B6, and folic acid. It's the most number of numerous cells in the body. It circulates for approximately 4 months before recycling, and several million are produced each second. Hemoglobin in RBCs transport oxygen from lungs to peripheral tissues, carbon dioxide from tissues to lungs. Yeah. So I'll share the screen again. Hopefully this will not happen again. I hope I'm audible and my screen. I hope I'm audible and my screen will be visible. Give me 2 minutes. Yeah, so this is what we're talking about. Now, what do you mean by denaturation? So, protein denaturation really happens when a protein changes its shape, usually uncoiling, and it changes its function and properties, and an egg is mostly liquid until cooked milk becomes yogurt or cheese when acids add like this, and heats, acids, bases, alcohol, heavy metals, enzymes, or other agents can cause denaturation. What do you mean by protein degradation? So, stomach releases HCl which denatures protein strands. It converts the inactive form of pepsinogen into the active form pepsin. Now, pepsin breaks the proteins into smaller polypeptides, and pepsin is one of the thousands of enzymes which allows chemical reactions to take place in the body without being affected itself. Now, polyintestine releases pancreatic and intestinal proteases, we know that, and basically they hydrolyze the polypeptides further into tripeptides, dipeptides, and finally amino acids, which are actively transported into cells and then into the bloodstream. This is basically the protein dilation in the GI tract. I'll not go into details. How are proteins absorbed though? They have carrier cells of the villi of the small intestine which have gates through which carrier substances transport to the amino acids. Capillaries are the smallest branches of the circulatory system that carry the free amino acids from the villi throughout the body. Okay. How are proteins synthesized? DNA in the cell nucleus gives mRNA the instructions. mRNA, basically the transcript, goes into the cellular fluid and attaches itself to ribosomes. To transfer RNA carries free amino acids to the mRNA. Ribosomes move along the mRNA, allowing enzymes to bond one amino acid to another until the completed protein is finished and released. This is about protein synthesis. The DNA serves the template to make strands of mRNA. Each mRNA strand copies exactly the instructions for making some protein the cell needs. The mRNA leaves the nucleus through the nuclear membrane. DNA remains inside the nucleus. And the mRNA attaches itself to the protein making machinery of the cell, the ribosomes. Another form of RNA, transfer RNA, collects amino acids from the cell fluid. Each tRNA carries its amino acids to the mRNA, which dictates the sequence in which the amino acids will be attached to form the protein strands. Thus, the mRNA ensures the amino acids are lined up in the correct sequence. Now, as the amino acids are lined up in the right sequence, then the ribosomes moves along the mRNA. An enzyme bonds one amino acid after another to the growing protein strand, and the tRNA are free to return for more amino acids. When all the amino acids have been attached, the completed protein is released. Finally, the mRNA and the ribosome separate. It takes many words to describe this event, but in cell, 40 to 100 amino acids can be added to a growing protein strand in only a second. So when a cell makes a protein, it is said that the gene is expressed. Nearly all the body cells can make all human proteins, but each type of cell makes only the kinds of proteins it actually needs. So what will be the role of proteins? It can be a building material, like for growth. A matrix of protein underlies almost all structures in the body, including bones, muscles, ligaments, tendons, connecting matrix between cell walls, scar tissue, hair and nails, maintenance, gastrointestinal tract. Cells are replaced every 3 days. The whole body has its cells renewed every 7 years. The enzymes are usually composed of a protein, a vitamin, and a trace mineral. They act as catalysts, allowing reactions to occur more quickly and efficiently. They can cause two substances to come together, making a new structure, or can split a compound apart. An enzyme is not affected by the chemical reactions it allows to take place. Now, if you know the role of proteins, it can be enzymes. Basically, they can be biocatalysts, they can help a reaction to happen without itself getting changed in the process. That's what enzymes or biocatalysts are. Now, roles of proteins can be growth hormone, insulin, glucagon, thyroxine, calcitonin, paras hormone, antidiuretic hormone, and all of that. So, hormones are nothing but chemical messengers that are secreted by endocrine glands in response to altered conditions in the body. Each travels to one or more specific target tissues or organs where it elicits a specific response. Proteins can also be used in the regulation of fluid balance. Independent edema may be caused when there is too much fluid between cells and not enough hydrophilic protein within the cells. Whereas acid-base regulation, proteins act as buffers, accepting and releasing hydrogen ions, thus preventing acidosis or alkylosis. Then we have transport proteins, basically which picks up sodium from inside the cell, changes shape and releases sodium outside the cell. Transport protein picks up and potassium from outside the cell and changes shape and releases potassium inside the cell. Proteins in immunity. Antibodies are giant proteins that bind up specific invaders like viruses or antigens. Antigens are substances that cause the body to produce antibodies. They may include bacteria, allergens, toxins, or anything that causes an inflammatory response. Now, Proteins, they are the source of energy. The brain and the nervous system must have glucose. Once the amino group is removed from the protein, the remaining carbon molecules can be used to create energy, 4 kilocalorie per gram, or stored as fat. Other roles include being converted to other proteins or making neurotransmitters, norepinephrine and epinephrine, melanin, fibrin as precursor of the vitamin niacin. Now, roles of proteins, other roles include being converted proteins to other proteins or making neurotransmitters, melanin, fibrin, precursor to the vitamin niacin, all of these. Protein turnover is basically amino acids pool includes free amino acids from endogenous or exogenous sources, nitrogen balance, positive nitrogen balance during growth or when negative or building new tissue. Negative nitrogen balance if burn, fever, injury, infection, or starvation, and all of that. So, using amino acids to make proteins, cells can dismantle one amino acid and combine the amino group of that amino acid with carbon fragments from glucose metabolism to make another essential or non-essential amino acid needed. So, deamination is the removal of the nitrogen containing amino group, converting it to ammonia, which is sent to the liver and converted into urea. The remaining carbon fragment may be burnt or stored as fat. Amino acids can be used to make fat by removing the amino group and converting the remaining carbon atoms to carbon fragments to fat. Okay. Proteins are degraded and resynthesized continuously. Several times more protein is turned over daily within the body and then is consumed. So, consumed amino acids in excess are unable to be used and not stored. They are degraded into urea, uric acid, or creatinine, lost in feces, urine, or sweat, and can be converted into hair and nails as well. Okay, so we won't discuss about amino acids too much at the moment. We have discussed it a lot in previous class. So yes, you were right. The I'm going to talk about, yes, the tryptophan is an essential amino acid which is also an aromatic amino acid. Okay, and it's both glucogenic and ketogenic. It can synthesize niacin, a vitamin of B complex group. It's a heterocyclic amino acid and chemical is alpha amino 3 indole propionic acid. It's only amino acid with an indole ring. Okay. We'll not discuss about the metabolic fate. If needed, we'll come back from another question. Phenylalanine and tyrosine are also the other two aromatic amino acids that need to be discussed. We have discussed all of these in previous classes. Not going in detail. So coming back to the question, the amino acid residues which are aromatic are phenylalanine, tyrosine, and tryptophan. Anybody has any questions? I'll wait for a few minutes and we'll move on to the next question for today. Okay. So we'll move on to the next question for today. Non-essential amino acids, we just discussed this. So you should be able to answer. Non-essential amino acids are, these are the four options. A. Synthesized in the body. B. Not synthesized in the body. C. Obtained from diet. D. Threonine, leucine, valine. Anybody? What are non-essential amino acids? A. Synthesized in the body. B. Not synthesized in the body. C. Obtained from the diet. D. Threonine, leucine, valine. Which of the option is correct, and please explain. I'll wait for a few minutes and I'll move on to the discussion. Anybody? Which one do you think is the right answer? Hello? Yeah, right. So yeah, non-essential because it's not essential to be taken in your diet. So we have talked about all the amino acids. I'm going to skip the previous parts. Yeah, these share the aromatic R groups, phenylalanine, tyrosine, and tryptophan that we were talking about. Now we can, as you know, if we do the classification based on nutritional requirements, we can divide it into essential amino acids, semi-essential, which are essential amino acids can't be synthesized in the body. That's why they're essential to be taken in diet. Their deficiency can affect growth, health, and protein synthesis. Semi-essential are formed in the body but not in sufficient amount for body requirements, especially in children. So arginine and histidine can be termed as semi-essential, whereas non-essential amino acids are basically they are synthesized in the body, so they don't have to be supplied from food under normal conditions. Like for example, alanine, aspartic acid, glutamic acid, all of these. Okay, so they are basically made by your own body from other precursors. So you don't need to eat them to survive. Though you often still do in your normal diet, but it's non-essential in your diet. Okay, so that's what non-essential amino acids are. So what are some other functions of amino acids? Apart from being the monomeric constituents of proteins and peptides, amino acids can also serve a variety of other functions. Like some amino acids can converted to carbohydrates and are called as glucogenic amino acids. Specific amino acids give rise to specialized products like tyrosine, tryptophan, which forms hormones such as thyroid hormones, epinephrine and norepinephrine, and a pigment called melanin. Tryptophan can synthesize a vitamin called niacin, glycine, arginine, and methionine acid. Sweetening glycine and cysteine help in synthesis of bile salts as well. Glutamate, cysteine, and glycine synthesize glutathione. Histidine changes to histamine and decarboxylation. Serotonin is formed from tryptophan, and glycine is used for the synthesis of heme. Pyridines and purines use several amino acids for their synthesis, such as aspartame and glutamine for pyrimidines and glycine, and aspartic acid, glutamine, and serine for purine synthesis. Some amino acids, such as glycine and cysteine, are used as deoxyribonucleotides of specific substances. Methionine acts as active methionine, like S-adenosine methionine, and transfers methyl group to various substances by transmethylation. Cysteine and methionine are sources of sulfur. So I've already discussed about the amino acids, and I am not going to go into detail again and again. So basically you are talking about the nutritional classification. Okay, one thing, what are standard amino acids? The amino acids that are used to form proteins, that is recognized with the ribozyme machinery or the amino acylation system, like histidine, isoleucine, leucine, lysine, threonine, tryptophan, methionine, phenylalanine, valine, glycine, etc. Non-standard amino acids are these amino acids that are not required to build proteins. They have a vital role as metabolic intermediates like hydroxyproline, hydroxylysine, carboxyglutamate, diaminopimylate, all of these. So these are the derivatives of amino acids and have role in metabolism, like alpha aminobutyrate, right? Citrulline, ornithine, which is involved in urea cycle, beta alanine, etc. Now I've talked about the nutritional classification, like essential, which cannot be made by the body. As a result, they must be taken from foods such as arginine, histidine, isoleucine, leucine, lysine, methionine, alanine, threonine, tryptophan, and valine, whereas non-essential ones are made by humans and is not essential in the human diet. They can be alanine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Okay, so that's the difference. On basis of number of amino and carboxylic groups, they can be classified monoamino, monocarboxylic amino acids, like aspartic acid and glutamic acid, diamino monocarboxylic amino acid like lysine, arginine. So monoamino monocarboxylic because there is one amino group and one one second. Monoamino and monocarboxylic because there is one amino group and one carboxylic group. Both are mono. Monoamino dicarboxylic because there is one amino group but two carboxylic group, right? One amino. Then diamino monocarboxylic because there are two amino groups here and one carboxylic group. We have talked about the physical properties. We have talked about chirality in one of the last classes. Amino acids will undergo decarboxylation to form the corresponding amines. They can be like histidine to histamine, tyrosine to tyramine, lysine to cadaverine, all of these. Okay. Now, what happens with anhydrine? So, when anhydrine reacts with alpha amino acid, this is formed hydrine and hydrine and aldehyde. Okay, and that's what we basically check in our experiments. What happens with the reaction in alkali salt formation? The carboxyl group of amino acids can release a H plus ion with the formation of carboxylate ions. Then the amino acids react with alcohol to form ester. The esters are volatile in contrast to form amino acids. Then diacetyl chloride means dimethyl aminosulfonyl chloride. When the amino acid reacts with diacetyl chloride, it gives a fluorescent diacetyl derivative. When the amino acids react with acid chloride and acid anhydride in alkaline medium, it gives thionyl amino acid. Then we have reactive Sanger's agent, Edmonds agent. Okay, so basically in Sanger's reagent reacts with alpha amino acid to produce yellow color derivative DNB amino acid. Whereas Edmonds reagent is called phenyl isocyanate. When amino acids react with Edmonds reagent, it gives phenyl hydantoic acid. Finally, it turns into cyclized form, phenyl hydantoin. Coming back to the question that we started with, non-essential amino acids are synthesized in the body. You were right. Anybody has any questions on this? I'll wait for a few minutes and we'll move on to the next question for today. Okay, so going to the next question for today. Which of the two amino acids possess an overall negative charge in their side chain groups at physiological pH? I'll repeat the question. Which of the two amino acids possess an overall negative charge in their side chain groups at physiological pH? Option A, cysteine and methionine. Option B, arginine and histidine. Option C, aspartic acid and glutamic acid. Option D, proline and valine. Anybody? Which one do you think is the right answer? Anybody? Okay. So basically, there's one option here that is correct, which will be aspartic acid and glutamic acid, right? Why? I'll go to the structure. So if you see, not over here, over here, but I think it's not visible properly. Wait, amino acids with uncharted polar chains. Yeah. So amino acids, aspartic and glutamic acid are proton donors, right? So at physiological pH, the side chains of these amino acids are fully ionized, containing a negatively charged carboxylate group. So they are therefore called aspartate or glutamate to emphasize that these amino acids negatively charged as physiological pH. Right? So at physiological pH, these exist in their mostly in the deuterionic form, meaning the amino group is protonated and the carboxyl group is deprotonated. So the side chain determines if the amino group has overall amino acid has an overall positive, negative, or neutral charge. So aspartic acid side chain has a carboxylic acid group with a pKa 3.9. So at pH 7.4 it is deprotonated, negative charge. Glutamic acid side chain has a carboxylic acid group pKa 4.1. So at pH 7.4 it is deprotonated, negative charge. So these two are the acidic amino acids and they carry net negative charge in their side chains at physiological pH. The other options are incorrect because cysteine and methionine they are neutral at physiological pH. Cysteine has SH group, methionine has a thioether group. Arginine and histidine are both basic amino acids with positive charge tendencies. Arginine always plus 1 at pH 7.4, histidine partially plus depending on the environment. Proline and valine are both again neutral, non-polar amino acids at physiological pH. Okay, so that's why the correct answer will be aspartic acid and glutamic acid for this question because they are acidic side chain containing amino acid. Okay, let's see if I have a better image of these two. Please, yes. So at 7.4 pH, this is plus, this is minus, so that makes it neutral. But the side chain is a carboxyl group which is negatively charged at physiological pH. So aspartic acid is negatively charged. Similarly, the same thing for glutamic acid. So both these amino acids are negatively charged at physiological pH. So coming back to the question that we started with, the answer will be aspartic acid and glutamic acid. Okay. So this was the last question for today. If you have any doubts regarding this question, please ask, or we'll stop the session today and meet again next Monday, same time. Anybody? Anybody? Okay. If nobody has any doubt, we can stop this meeting and conclude it today. My laptop is showing some issues also as well. So by next class we can meet again next Monday. Yes, yes. Sorry, your voice is cracking. Can you write? Okay, thank you. Sorry. Can you write in the chat box? Your voice is cracking a bit. Okay, thank you. I'll upload all the videos, so you don't have to worry about it. We'll mix again next Monday. Thank you.